Okay, so we're going to call this the ribosome action in detail, or in more detail. And this is sort of the AP portion of this. We want to have a slightly better understanding of what's going on in there. Okay, so first of all, the ribosome actually has two separate sort of sections to it. So, not sure what happened there. Hang on, I'm back. Still back. Okay, I must have touched the cord. Perhaps it needed to catch a few winks, go to sleep. I don't know, something weird. So, that's our title. So the ribosome comes in sort of two parts. It has a large subunit, and then it has sort of a small subunit. So let's call it the large subunit and the small subunit. And essentially, these are divided by the different active sites that they have and the different sort of things that they bind to. So remember, we're drawing these sort of weird pictures. This one looks like a messed up derby hat from 1912. But what these are lumps of molecules with atoms and electrons and everything. And of course, we know about binding sites and how as molecules come together, those things are interacting all the time. So as we get on in biology, we start drawing sort of these representative shapes. But always remember that this shape is a bunch of atoms. And those atoms interact. And the distribution of electrons is what basically causes these active sites to interact with each other. Okay, so we're going to start with the small subunit first. All right, so the first thing that happens is the small subunit, we'll draw it on the bottom, it has sort of three sites. I'm going to label the sites in green. Over here, and actually in the textbook they draw it sort of like almost like a flat sort of donut with a dent instead of a hole, kind of like a red blood cell sort of, but we're not going to get too crazy. So we'll have right here, we'll call this the exit site. This is the place where the developing, where the messenger RNA is sort of on its way out, okay? We'll talk about it in a second. Then we have the next little spot here. It's called, we'll call it the P site. So this is called the E site, exit. This is the P. And then this is the A site, like this. All right. So the P and the A stands for the location site. This is where what we call the aminoacyl. I'm going to get a smaller thing here. Aminoacyl transfer RNA. So that means it's a transfer RNA that has a protein attached to it, essentially, or amino acid, I'm sorry. It's got an amino acid attached to it. So it represents the amino acid and the RNA sort of together as a single unit. Okay, so let's go through the process. The P site is usually where we see the attachment of the starter codon, which technically is methionine, the amino acid that stands for start. If you go back to your book on the chart, you'll see that AUG codes for methionine, and that is the start codon. It's the amino acid amino acid transfer RNA complex that stands for start. Okay, so here's what happens first. AUG, methionine, stands for start. Record. All right, so the P site now is the peptidyl site. So This is the aminoacyl site. This is the peptidyl site. And you'll see what all these mean in a minute. Peptidyl, YL. All right. So now what essentially has to happen is we have to get the messenger RNA to bind with this. So there has to be a process, right, which this has to happen. And it has to sort of bind in the appropriate way. So in step 1, we're going to get the start codon showing up sort of first. So the first thing that sort of happens, and I'll do this in stages, and I guess I can draw the transfer RNA, something like that. This is the start codon. So it's going to come into place here. Let me put some little arrows showing that it's sort of binding at this site. And it's carrying methionine as its amino acid. So we'll do the amino acid in red. It's carrying behind it methionine. And methionine is important because it's the go codon. It's the starting codon, right? So that's going to be binding in this site. So this is going to be our scanner. That's what we're setting up here. We're setting up a scanner. So the next step then is once that binds, let's go down and draw a second picture. So we'll draw that sort of thing again. We'll put in those 3 binding sites. The exit, the peptidyl, and the aminoacyl right there. And now we have this methionine that's being held here. Now, it's being held in this place. I guess I should go back. It's not being held by the codon. It's being held in place by the larger subunit. The larger subunit is sort of around the back. Well, we'll get to that in a minute. So next, let's draw that little guy in again there again. So here he is. There's his codon, or anticodon actually. And he's got methionine on the back of him. So that what happens then is this unit is going to ride along the messenger RNA molecule. It's going to start at the 5 cap. So the messenger RNA molecule is going to come in here. I'm going to draw that with the sort of the blue. Here's the long messenger RNA molecule. It's going to come through here like this. It's going to start at the 5 cap. So remember, there's a little section here. I'll put it in yellow. And that's the 5' cap. And it's the enzyme. The enzyme is, the ribosomal enzyme complex is going to travel along this until it finds or reads the proper sequence on here. Now, the anticodon on this methionine is UAC. UAC, which means the blue codon that it would read would be AUG. AUG, which is the codon for methionine, which is the codon for start. So what it's doing is it's going along this molecule and it's using this transfer RNA that's held in place to scan and find the starting point. It's looking for the matchup for the AUG, looking for that sequence. And when it finds it, that's the starting point. Once it finds it, that allows the larger subunit to sort of bind in place. So what we're going to do is we're going to draw the larger subunit in black, sort of kind of around here like this. So let's write beside here sort of a little description of what's going on. The ribosome. The ribosome is going to move along Oh, I don't like this thing. The ribosome moves along the messenger RNA until it finds the AUG codon, which is the start codon. So this is called it's scanning. That's what we call this. It's scanning this molecule to get ready to go where to start. So I guess what we should write on this side, perhaps, is we should write the first step is that the transfer RNA, the tRNA for methionine binds first. Because that's what basically, that's the scanner, right? So then you run this thing through, oh, right there, it finds that particular section. So we're sort of like scanning. So the first thing that happens is this, and then this runs along until it finds that. Okay. Say that what? Okay. So this large ribosomal unit is moving along the messenger RNA in this direction. And what it's doing is it's moving along. So the blue piece is sort of going through, being fed through the ribosome. And it'll come out of the ribosome at the exit site and dangle off into the air or into the cytoplasm. And until this methionine little scanner locates the AUG start spot. And now it knows that's the beginning, yes. So we've essentially located the start. Next picture. All right. Let's draw again. Let's go down below here. There's our small subunit and the exit site, tidal site. All right, and aminoacyl site. Good. Okay, so we have those three sites. And now the methionine is sort of locked into place here. Oh, you know what? I'm going to leave a little more space in between there. Hang on. So we can draw the messenger RNA better. So let's go like this. Okay. And of course it has the methionine attached to it. Methionine. The blue RNA is sort of moving through, right? Yes. It's now sort of coming out the exit site, and we've found the start codon. So what happens is that methionine is bonded or is held in place. And then there's another little guy that comes along. Let's use purple, I think. And this is a little peptidyl transferase. So watch what happens here. This little guy is an enzyme. His name is peptidyl transferase. And what he's going to do is he's going to detach this methionine from this amino acid, but he's going to have attach it to something. So he's getting ready, all right? So now what's happening is the next one is coming into place here. So at this site, we have another one lining up now, and it's pulling another amino acid. It doesn't really matter what one it is. So at this point, I'm just going to put AA, okay? Because methionine is important because it's the start. But this could be any amino acid that comes next, whatever's next in the chain, whatever the code's for. All right? We could draw little lines here just to show that these are sort of being these codons are being attached. Now, this peptidyl transferase is going to detach the methionine from this transfer RNA and move it over and attach it to that other amino acid. Okay? So peptidyl transferase, I guess if that's a before picture, here's the after picture. What I'll do is, here's what I'll do. I'll take this section right here, and I'm going to draw it over here, sort of after what happens. So we don't have to redraw the whole picture because it's just this section that's affected. So here's the first guy, and now here's the second guy, second transfer RNA, and what happens is his amino acid is still there, and it's attached now up here. The methionine is translocated, it's moved and attached there with a peptide bond. So there's your methionine, and this is the other amino acid. And so now it's free. It's free from the transfer RNA that was there. Okay. And now you can see what's going to happen is this process is going to repeat itself. So the ribosome is moving along the RNA molecule. Remember, the ribosome is sort of moving in this direction. And so each time it moves a little bit, a new transfer RNA could come into this position right here. There's another codon waiting. All these codons are waiting to be lined up. So these transfer RNAs will continue to come in, and each time they do, the peptidyl transferase will pull the amino acid off of the transfer RNA and attach it onto the other amino acids waiting. So a picture of much later on, if we go down to sort of a much later on picture, we've got this. We've got the exit, the P, and the A. I suppose you are still drawing this one, aren't you? Okay, we'll pause for a second. So let's carry on a little bit later. Oh, you know what I didn't draw up here on the other picture, I just realized, is the larger subunit, which would be sort of around the back there. That's kind of important because that has to be there for this to happen. So don't forget the larger subunit. In fact, if I took a color, like a light gray here, maybe a crayon that's light gray, and I shade it a bit, there's the larger subunit. Okay, that circle I drew is really just a visual to show you what happens. And then here's the other subunit. We'll kind of shade them in a bit so you can see them. Okay. So now we have this subunit, and we're going to have to draw okay. So the messenger RNA is moving through, and it's leaving at the exit site. So now we've read along several codons, right? We're still reading codons, but maybe we've read along 10, 12 codons by now. And so we have the, as it moves along, we have the next amino acid coming into play. So we get this, okay, so the methionine was transferred. So now the sort of empty transfer RNA that held on to the methionine moves over to the exit site. It has no amino acid attached to it because that amino acid has been taken off. That was the methionine. And that means that the other one now moves into this position. And it's carrying the amino acid that it brought, and also the methionine that was attached to it. So you see how everything is shifted over to the methionine. This would be whatever amino acid came next. And now another one's going to come along. But the amino acid, or the transfer RNA over on this side, he's at the exit site, which means boom, he's going to pop out. He's gone. So shortly after he's done, he exits the complex, because the exit site means that his bond is no longer strong at that point as it's moved along, and he just comes out. So now this guy's here. And then, of course, you're going to have another one coming along, and it will carry with it another amino acid. It doesn't matter which one anymore because it's just whatever's next in the sequence. And there will be the little peptidyl transferase that's moved along as well, right here. Peptidyl transferase. And what he's going to do, let's put the large subunit in. Let's color it sort of grayish. I don't know if that's my gray or not. No, this gray. Right, there's the large subunit. Okay. So now I'm going to do the same thing. I'm going to draw a little circle around that top part. Come on, board, answer me. I don't know what's happened now. Hang on. It's not, there it goes, pen. Okay, so we'll get a little circle here, right here, and we'll bring it out here, and we'll have a look at what that looks like or what's happening. So now you'd have the middle transfer RNA would be sliding over to the exit site, right? And this one would be sliding over, and we would end up with the first one would be completely gone. The second one is sort of on its way out. He's lost his amino acid, and then the other one is moving over into the middle section, and it would have now the two amino acids. See the MET, the methionine, and the other amino acid? They're going to be linked, and so we'll get three little boxes here. Like this, detach them, and of course, this top one is the methionine, the starting one, and these are just other amino acids in the sequence. And you can see what's happening. We're starting to build a chain of amino acids that would start to exit or hang out of the ribosome as well. So afterwards, when you get near the end of all this, I guess I better let you catch up to me first, or you're all going to ask me to go back. Okay, so if we draw, let's draw it way down here now, because it's moved along. So there's our ribosomal unit, right? We can put the three sort of binding sites there, the exit site. There we go. And so now we're almost at the end of our piece of messenger RNA. See, it's moved all the way to the end. And so what will happen is there will be a stop codon. The stop codon is a little bit unique in that when it binds, a new transfer RNA doesn't fit. It doesn't bind in there. And so what you get is another little protein. It's a protein, what do they call it? A factor, protein release factor. That's what they call it. And it'll bind in there. So the very last transfer RNAs are in place here. That one has lost it. This one is still getting its last piece here, right? And what's happened, of course, is rather than draw the boxes, what we've built or assembled here is a very long chain of amino acids. So those are the red boxes, short form, right? And then you get this this binding of. I don't want to use purple because I've used that for something else, and I don't want you to think it's the same thing. So we get another color here, nice reddy brown color. And this is sort of what happens when it's time to stop. So this protein release factor gets attached. That says protein release factor gets attached. Instead of another transfer RNA. Okay, instead of. I'm not writing all that down, but you can add it in. And this ends the chain. Because if there's no more transfer RNA, there's no more amino acids, and then the chain is released. And so what we have up here is our polypeptide peptide chain that's being released. So this ribosome is a pretty complex thing. It's a complex bundle of ribosomal RNA and proteins of various kinds. And the complexity of the bundle allows it to have these complex active sites that can hold these molecules sort of in place and create this scanner that can read until it finds a start codon, and then have a sequence of transfer RNAs sliding into place, and then the amino acid chain being sort of separated from them and fused together, and then like a long string tangling off of this whole thing. And I think what I'll do is see if I can find you some actual videos now that we are finished with our pictures. Question?